Hello, everyone. Happy New Year. I want to thank the organizers for the invitation to speak, and I also want to thank Chris, Giovanni, and Liama for organizing this series, which has been important to keep the community connected until we can reinitiate sort of normal type meetings. Hopefully sometime towards the end of this year, we'll be able to actually see each other in person again. So the topic of my talk is the orchestration of the Sphingolipid Metabolic network. And over the past 150 years since sphingolipids were discovered, basically all of the lipids and the enzymes, the genes, how the cell biology works, and many of the important functions have been identified. And we know that the pathway is pervasive in biology. Something that's less understood, I think, is how the overall pathway is regulated. We know a lot about how some individual enzymes are regulated, transported, how lipids are transported, and so on, but how there's coordinate regulation of the pathway to ensure its proper function is less well understood. So my kind of simplified drawing of the sphingolipid pathway kind of resembles a metro line. And this is the, in case you don't recognize it, this is the Washington Metro line. And our NIH is actually at Medical Center, which is here. Capitol is down there, so we're quite a distance away from where all the action was last week, thankfully. But just like a well-functioning metro line, it has to be, the pathway has to be regulated. For instance, like on a metro line, you have to have enough trains present to transport passengers to their destinations, but you can't have too many on the lines, otherwise they'll run into each other and there'll be problems. Under circumstances where you need a lot of, to cover a lot of people or transport a lot of people, there has to be some expansion of the metro's capacity, i.e. more trains on the lines, which might happen during rush hour or on special kinds of holidays. And finally, when you put the trains on the tracks and they're finished, you want to kind of recycle the trains back again. You don't want to constantly have to make new ones. That would be very inefficient. So in the sphingolipid pathway, at least this kind of stripped down version that I'm showing, there are several key features. One key feature is that there are bioactive intermediates that need to be made in order to produce the final end products that are important for the sphingolipid functions. And one of the key features of the pathway or key functions is the production of plasma membrane lipids, usually in the form of sphingomyelin or glycosphingolipids. And hearkening back to Tony Fuderman's talk, these are key molecules for fine-tuning the lipid bilayer. And also there was a talk about GM3 by Dr. Inokachi, who stressed the importance of the head groups of the glycosphingolipids for controlling receptor protein function. As I mentioned, there's another key component of the pathway is the production of bioactive lipids. And the most well studied of these lipids are sphingosine, ceramide, and sphingosine 1-phosphate. It's generally accepted that sphingosine and ceramide are pro-apoptotic lipids, and sphingosine 1-phosphate is a extracellular signaling lipid that engages a family of G protein coupled receptors to affect signaling in multiple physiological systems. And Tim Allah had given us a nice talk outlining all of this. As far as physiological functions, I'll mention a few. One place that sphingolipids have key importance is in the nervous system. The brain and the central nervous system is very rich in lipids, and in particular sphingolipids. In the white matter of the brain, which is composed of myelinated axons, the myelin, of course, is the insulator for the axons. 50% of the myelin is, or maybe more, is sphingolipid. These are mainly cerebrosides, galcir or sulfatide. In the gray matter, which are basically the neuron bodies in the glia, about 10% of the content of the neurons are gangliosides. So, many of the diseases of the sphingolipid pathway affect the nervous system, probably because it's so enriched in sphingolipids. Another tissue that's enriched in sphingolipids is the skin, especially the stratum corneum, which is lipid-rich, up to 50% ceramide, and this forms the epidermal barrier layer that's key for life, and it's produced by the production of the ceramides that are special ceramides that are omega hydroxylated and derivatized with linoleic acid, and form lipid lamella in the outer layers of the skin that are important to produce the barrier layer that's essential for life. Now, an illustration of how important it is to keep the pathway in check is from the many genetic diseases that are caused by mutations in the individual enzymatic steps of the pathway. And Konrad Santoff and Fran Platt have already talked about the lysosomal storage diseases. And I have those illustrated here, which involve the stepwise degradation of sphingolipids in the lysosomes. Not only are these important in these individual genetic diseases, but as Fran had noted before, there seems to be an overlap with some more common genetic diseases, such as Parkinson's disease. And this list of diseases has now expanded to the synthesis pathways, where there's now a disease at almost every step. And many of these diseases have a neurological component, presumably because of the importance of the sphingolipids in the nervous system. And some also have affect the skin, presumably because of the importance of ceramide in the skin. So I'm going to talk about 3 issues in relation to the Sphingolipid Metabolic Network. The first one is, how are the amounts of the bioactive intermediates organized with respect to the final products? How do we keep these in balance? The second is, how is the pathway coordinately regulated when there's a need for large amounts of perhaps sphingolipids. And finally, what is the significance of the recycling pathway? So let me introduce the ORMs, which you've already heard about from Tila Wargol. These were initially discovered in yeast by Breslow and Weissman. In mammals, there are 3 homologs that are very similar with greater than 80% amino acid identity, such that it's very hard to find an antibody that will distinguish between the 3 forms. As Binks-Wattenberg has shown, they interact with ceramide to negatively regulate the SPT complex, which is a 3 subunit complex of SPTLC1, 2, and a small subunit. And their role in physiology was not known. So when Ben Clark was in the lab, he proceeded to knock out the 3 ORMs, ORM1, ORM2, and ORM3, to try to understand the physiology and how they maybe to get some insight into how they work. We found that the individual knockout mice were all viable, seemed pretty normal. The double knockout mice were all viable. So we did an experiment where we tried to derive or see what the effect would be if we went beyond this to look at triple knockout mice, for instance. So in order to do this experiment, we set up crosses between triple mutant mice, ORM1 knockout, ORM2 het, ORM3 het, which would give us all of these predicted genotypes. And after quite a period of time and obtaining more than 100 pups, we could quantify what was observed and compared that to the genotype that was predicted. And what we found was, although, let me see if I can collapse this. If we look at the box with no alleles, we would have predicted that there would be about 10 mice, and we found 0, suggesting that this is a lethal at some point. And to go beyond that, if we look at the offspring that would have 1 allele, again, we found 0 mice that were ORM1 knockout, ORM3 knockout, ORM2 het. And the class that would be ORM1 knockout, ORM2 knockout, ORM3 het, we found just 5 where 20 would have been predicted. So this suggests that the ORMs are, at least in combination, are required for postnatal life. Now, this genotyping was performed after weaning, so we don't really know at what point the lethality occurred. So we looked at or focused our analysis on the single knockouts and the double knockout mice, and looked at lipid levels in a variety of tissues. And the tissue I'm showing here is brain. So we have the 3 single knockouts, ORM1, 2, and 3, and then the double knockouts, 1, 2, 1, 3, 2, 3. As far as the single knockouts go, we saw that in some cases there was, in the ORM3 knockout, there was a somewhat of an elevation in some of in ceramide and sphingosine, perhaps dihydrosphingosine. In the double knockout mice, the biggest effect was seen in the ORM 1-3 double knockout mice, where there was a very large increase in all of these intermediates. Looking at just the gross phenotype, we were able to notice pretty quickly that just by body weight, the ORM 1-3 double knockout mice were distinctive compared to the others, which is going along with what we saw from the sphingolipid levels. And let me just show you a brief movie to illustrate the sort of phenotype we saw. So the ORM 1-3 double knockout is on the left, and you can see that it has this distinctive clasping kind of behavior, indicative of a neurological problem. And in this sort of cage walk, although both of the mice aren't doing too well, I think the double knockout has more of a problem and is unable to navigate the cage very well. Go to this one. So to sort of quantify some the neurological aspects of the deficit, we did a wire hang test, which measures the strength and coordination of the mice. Basically just hanging the mice from a wire coat hanger by their front limbs. And we found of all the groups, again, the 1-3 double knockout mice performed the worst. They were different from all the others and showed a clear deficit. So in trying to figure out what exactly was wrong, our thoughts immediately went to the importance of sphingolipids in the central nervous system, and the fact that we could spot the deficit sort of early in life. I think those mice were 6 or 8 weeks old. Led us to focus on perhaps the myelin being an issue, because during development in mice after birth, the myelin membrane expands considerably. Myelination occurs. So there would be an enormous amount of sphingolipid synthesis needed to produce this myelin. So for the analysis, we went to the sciatic nerve because it's relatively easy to do the histology to determine how well myelination is going. And so these are EM images from sciatic nerve of wild type, ORM 1 knockout, ORM 3 knockout, and ORM 1-3 double knockout. So I don't think you have to be an expert to notice immediately that there's an aberration or a problem in the 1-3 double knockout sciatic nerve. The axons are less dense in the field, and we quantify that here. And there's also many abnormal appearing figures of myelin, in contrast to the sort of the well-organized structure of the wild type. And many of these figures appeared in this, like these images in the bottom panel, which are showing redundant myelin, as if there is actually too much myelin being made by the Schwann cells. Analysis of the sphingolipids in the sciatic nerves showed a very elevated amount of sphingolipid metabolites, precursors, in the 1-3 double knockout mouse in the blue. And sort of paradoxically, there was a lower level of the final product, hexosylceramide. So our interpretation here is that without the ORMs, 1-3 ORMs, that releases the brake from the SPT, which allows it to produce large amounts of precursors that overstrip the capacity of the final enzymes to produce the hexosylceramide products. The lower level, we believe, is due to the fact that a lot of the Schwann cells are dying. You know, there's a lower number of myelinated axons in those figures. So we next wanted to ask the question as to whether it was actually the overexpression of sphingolipids that caused this myelination. It's been known that, you know, the absence of sphingolipid synthesis causes myelination problems, but I don't know that it's been ever shown that overexpression of sphingolipids caused a myelination problem. So to do this experiment, we used a nice tool that Teresa Dunn developed, and in order to overexpress the first step of sphingolipid synthesis, serine palmitoyltransferase. So she has been able to devise a single chain enzyme, fusion enzyme, consisting of SBTLC2 fused to the small subunit, which in turn is fused to SBTLC1, which is active and actually under control of the ORMs. We produced mice that have a knock-in version of this fusion SBT that's under inducible control by Cree, and bred those mice with a Cree mouse, inducible PLP Cree mice that express the Cree enzyme in Schwann cells and oligodendrocytes. And upon feeding the mice tamoxifen, we can affect the activation of the fusion SPT gene. So, and we got quite a dramatic result with these mice. So let me show you another little movie. So these mice were about 4 weeks old when we fed them the tamoxifen food, and this is just 2 weeks later. And they showed severe sort of hind limb paralysis just after 2 weeks of the feeding. It's to such an extent that they had to be immediately euthanized. And remarkably, the picture that we got when we analyzed the sciatic nerves of these mice was very similar to the ORM knockout mice, that there were in the activated fusion mice, SPT mice, there were lower axonal densities and an abundance of figures with these with this excess myelination, redundant myelination. And like the ORM knockout mice, we found that there was a large amount of sphingolipid intermediates, ceramide sphingosine, dihydroceramide, dihydrosphingosine, after activation of the fusion SPT, and without a corresponding increase in the hexosylceramide. So to summarize this part, we've shown that simultaneous deletion of ORMs caused bioactive sphingolipid increases, abnormal myelination, and neurological impairment in the mice, and that the elevation of SPT alone caused nearly identical phenotype and myelin abnormalities. So the ORMs function redundantly to control excess sphingolipid synthesis during myelination, and that the ORMs therefore orchestrate an orderly sphingolipid biosynthetic pathway, and that there's a balancing act that occurs between the production of the end product sphingolipids for myelin and the production of the metabolic intermediates, which could be disruptive if produced at high amounts. And myelination is particularly sensitive. I mean, that's basically the observation that we found to dysregulation of the pathway, presumably because there's such a high flux of sphingolipid metabolites moving through the pathway. So let me turn to the next part, which is how the sphingolipid pathway might be coordinately regulated. So this would be important in the example of myelination, for instance, where you might need to elevate the synthesis of an end product, but in order to do that, you might have to elevate the synthesis of the individual enzymes. So in order to look for such a regulator, we undertook a CRISPR-Cas9 genetic screen, and this was done in HeLa cells. And we used the so-called human gecko library, a genome scale CRISPR knockout library, which is a pooled lentivirus library that targets over 19,000 genes with about 6 guide RNAs per gene. And we selected for surviving cells with Shiga toxin. So we weren't the only ones to do this. There's actually been several papers published with a similar screen. We were kind of the last to publish it, but unfortunately, but in almost all cases, very similar profiles of genes were discovered. So that was nice. So Shiga toxin, of course, is the bacterial toxin, which consists of 2 subunits, an A and a B subunit. The B subunit binds to its receptor on the cell surface, which is GB3. It's a glycosphingolipid with 3 sugars on it, glucose and 2 galactoses. And the way it works is it binds to receptors and internalized, makes its way into the ER. The A subunit enters the cytoplasm, which inhibits protein synthesis and kills the cell. So here's an illustration of the sphingolipid pathway in PowerPoint with the production of GB3, the Shiga toxin receptor, which goes to the cell surface, and the core enzymes needed to produce the GB3. So if the screen would work, we would expect to be able to find hits in all or many of these genes. So as a proof of principle to show that this approach would be valid, we knocked out SPT, which we would predict would remove the GB3 and make the cells resistant. Nicely for us, there's a very good antibody called CD77, which recognizes GB3 on the cell surface, which allows us to probe the expression. And in wild-type HeLa cells, we can see that most of the cells, in this case, express GB3. And when the SPT is knocked out, very few of the cells express it. So this is proof that modulation of the pathway would affect GB3 expression. So to actually do the screen, we took our HeLa cells that express Cas9, infected the cells so that we would only have one lentivirus infecting one cell, divided the cells in half and treated half with Shiga toxin, which killed most of the cells, allowed the survivors to grow up, and then isolated genomic DNA from the Shiga toxin treated survivors and the untreated cells to identify the guide RNAs that were present. Then ranked the guide RNAs based on their abundance and the relative number that we saw for each individual gene. And this is the top 20 hits. And in the hits, where there's all red arrows are all sphingolipid pathway genes. There's some other genes that are known to be involved in glycosphingolipid synthesis, like Golf3, which I believe Giovanni studies. And some of these genes have also been picked up in the other publications. Now, the one gene that we focused on, and this, so all in red here are the genes that we picked up in our top hits. But we were looking for possible regulators of the pathway. So our attention was focused on this gene, AHR. So what is AHR? So AHR is a transcription factor, the aryl hydrocarbon receptor. It's kind of an ancient transcription factor of the basic loop helix family, or the per-arnt sim family. It dimerizes with another protein, ANRT. It binds to response elements in promoters of responsive genes. It's probably best known for its exogenous ligand, dioxin, but there are also many endogenous ligands. It was initially known because it regulates xenobiotic detoxification responses, but now it's appreciated that it's involved widely in development and physiology. And just a little PowerPoint on how it works. It binds its ligand, whether that's a extracellular ligand or an intracellular ligand, dimerizes and activates genes that have the response element. So just to validate that GB3 is regulated by AHR, Soroff made helicell knockout cells for AHR, and we found that their GB3 expression was reduced. Ceramide levels showed that there was a modest decrease in the amount of ceramide. Pathway gene expression analysis showed there was a modest decrease in several of the important enzymes in the pathway, including one of the subunits of SPT small subunit A, also in the GB3 synthase, and in the glucosyl ceramide synthase. We looked at a number of these genes for functional response elements. I'll just show you the data from one, which is the small subunit of SPT. It has two predicted response elements. And by qPCR, we found that we could immunoprecipitate the DNA, the promoter DNA for this gene, compared with, say, a control IgG, showing that the promoter is occupied by AHR. We also use the knockout cells to look at the efficiency of the promoter expression, and using promoter constructs, we found that there was a reduction in the promoter activity for SPT SSA in the knockout cells, indicating that AHR was important for its full expression. To look at the physiology of the AHR knockout or the AHR gene, we made a knockout in mice of the AHR receptor. And ours wasn't the first knockout mice by any means, but it was probably quicker for us to do it this way with CRISPR. And we found that there were decreased sphingolipids in the mice, in the liver, and in the plasma, and in several tissues, and I'll show one here in lung. there was sort of a generalized decrease in the expression of a number of sphingolipid enzymes, including some of the ones we saw in the HeLa cells. So our next question was, does AHR regulate an important sphingolipid physiological process? And one that we're definitely familiar with was myelination, where there needs to be a high degree of sphingolipids produced for proper myelination. And so here's an analysis of relatively young mice, wild type in AHR, sciatic nerve. And we found that the G ratio, which is the standard way to sort of measure myelination, it's the ratio of the inner axonal diameter to the outer axonal diameter. So a larger number would be indicative of perhaps a thinner myelin. And we did see a significant increase in the G ratio in the AHR knockout, and actual direct measurements indicated that there was a thinner myelin in the AHR knockout, which is what you might predict if there was less sphingolipids being produced. And we looked directly at sphingolipid levels in the sciatic nerve of the AHR knockout mice and found that there were lower amounts of sphingolipids as well as lower amounts of sphingolipid metabolic enzyme expression. So to summarize this section, AHR is a positive regulator of sphingolipid levels through enhancing the expression of multiple genes in the biosynthetic pathway. AHR is important for full myelination, which actually had been shown before. So we weren't the first to show that, but it hadn't been linked to sphingolipid levels, which we believe is a part of this phenotype. Now, the final part of the talk is how the recycling pathway is regulated. Of course, by recycling pathway, what I'm talking about is the pathway where the sphingoid base that's derived from mature sphingolipids is recycled back into the pathway so that it can be utilized for synthesis of sphingolipids. And that's illustrated here. And generally, it's considered as recycling sphingosine that's derived from the degradation of sphingolipids in the lysosome that moving back into the ER and being converted back into ceramide. Now, sphingolipids are also in the extracellular space, especially S1P, which is important as an extracellular signaling lipid. So we were also interested in perhaps how that sphingolipid was handled by cells and whether it was reutilized in the recycling pathway. So we set up a screen again that used the Shiga toxin kind of paradigm. So we did the screen with the knockout SPT cells, which would lower drastically the level of GB3 on the cell surface. And then we added to the cells either S1P or sphingosine. So the idea is that the cells would incorporate this, the S1P or sphingosine and reutilize the sphingoid base for the production of GB3. And this experiment shows that what happens when we add sphingosine or S1P to the HeLa cells that are knocked out for SBT. So the knockout cells have very low GB3 level expression, but this is greatly enhanced when sphingosine or S1P is added. So the scheme for the screen is shown here, very similar as we did before, only this time after infection of the cells with the lentivirus library that carry the guide RNAs, we divided the cells into two parts. One part only had got S1P, and the other part received S1P plus Shiga toxin, with the idea that now the Shiga toxin will kill off any cells that produce GB3, and then deep sequencing to identify the genes. So I'm just showing the S1P screen here. So the list of top genes that we saw was similar to what we saw with the initial screen of the wild type cells, including UGCG, which is the glucosylceramide synthase, the glycosyl transferases to produce GB3 ceramide synthase. But we also saw two new genes. One was SGPP1, which is sphingosine-1 phosphate phosphatase 1, and PPAP2B, which is also known as LPP3. And this is a lipid phosphatase. So LP3 is an enzyme that's expressed on the cell surface. It hydrolyzes extracellular LPA as well as S1P. It was enriched in the S1P screening, but we didn't see it in the sphingosine screening. So it seems specific for S1P. It causes dephosphorylation of exogenous S1P at the cell surface, or perhaps in endosomes. SGPP1 is expressed, a big difference is that it's expressed in the ER. It also catalyzes S1P to sphingosine. It has been shown that sphingosine produced by SGPP1 is utilized for ceramide synthesis. And this was enriched in both the S1P screens and the sphingosine screens. So just showing that to validate or verify that LPP3 is expressed at the cell surface of the cells, maybe in some vesicles. Very different from SGPP1, which is expressed in the intracellularly in the ER. So clearly different locations. So now I filled in the positions of the two genes. So this would imply that the S1P must be dephosphorylated to sphingosine. But for SGPP to come into play, it must be rephosphorylated, presumably by sphingosine kinases. And if it's rephosphorylated, that would also be a substrate for the SGPL1, which is the lyase that irreversibly degrades S1P, takes it out of the pathway and puts it into other pathways. So to look at the relative importance of these enzymes in recycling, Lila and Marie did an experiment where they made knockouts of all of these enzymes, sphingosine kinase 1 and 2, including the double knockout, the phosphatase, SGPP1, and the lyase, SGPL1. And the way we looked in this experiment, the way we looked at recycling was to look at GD3 expression in wild type cells, and then GD3 expression of cells after extensive treatment with Mariocin. which we presume is the residual is the result of the fraction of sphingolipid synthesis, which is not affected by, which is not from de novo synthesis, so would be from recycling. And when we do that with wild type cells, we see a reduction of about 75% of GB3 expression. So presumably this is remainder is from recycling. And sort of what kind of validates that idea is that when we knocked out the phosphatase, we see complete inhibition of GB3 expression. Essentially, we're knocking out the recycling pathway with the phosphatase knockout. With sphingosine kinase 1 knockout, we see something not too dissimilar from wild type cells. With the kinase knockout now, we see less inhibition by myriocin, suggesting that perhaps there's more going into the recycling pathway. When we make the double knockout, though, we see very little. We're unable to significantly, or I guess it is significant, but substantially inhibit GB3 expression with myriocin. So this would suggest that actually the S1P isn't really needed for recycling, that even without sphingosine kinase, the recycling pathway is proceeding. So in the explanation for the, in that explanation fits with what we see with a knockout of the lyase. So knocking out the lyase, which prevents sphingolipid from leaving the pathway, renders the cells, at least under these conditions, unable to be inhibited as far as their GB3 expression by myriocin. So the results suggest that S1P from the cell surface, or perhaps sphingosine derived from lysosomal degradation of sphingolipids, is abundantly phosphorylated by sphingosine kinase to produce S1P, which occurs at a sort of a critical branch point in the pathway, which could either be acted on by the phosphatase to push it into the recycling pathway, or removed from the pathway by the lyase and be transferred to other lipid pathways. Just to go back to some experiments we did several years ago at the importance of the recycling pathway, now that we have a little bit of understanding of how it's working, we made phosphatase 1 knockout mice. Oops. And we found that the mice didn't live very long. They generally died after a few days. And their major phenotype was a skin problem, actually. So we saw that the skin would kind of peel off, and the stratum corneum layer would leave the mice. And if you remember, as I showed you, that the stratum corneum is highly enriched for ceramide. And keratinocytes from these mice showed lower levels, modestly lower levels of some ceramide species. So that kind of indicates that the importance of the recycling pathway in physiology. So to conclude this final part, we found that for extracellular S1P recycling, there's this dephosphorylation, phosphorylation, dephosphorylation cycle. And it suggests that a key role of S1P as a branch point substrate. It's not so much that the S1P is needed for recycling, because it looks like it probably isn't. Sphingosine could be recycled. But it's a point of regulation where a decision can be made to either maintain or remove sphingolipids from the metabolic pathway. And in physiology, it's important for the skin barrier, which requires large amounts of ceramide. Now, it's interesting that both of these enzymes, so these branch points are generally very important in metabolic pathways and subject to some regulation, because that regulation could determine whether the substrate is retained in the pathway or go into another pathway. I think there's very little known about how these enzymes talk to each other or are regulated individually, which could be very important for the pathway. So I'll end there, and I showed you a few ways that the pathway can be regulated. I think going forward, understanding the regulation is going to be important for perhaps devising therapies for diseases where levels are altered in for sphingolipids, whether these are the genetic diseases or more common diseases, which are now becoming more readily apparent. And let me thank my lab, Marie, Laura, Terry, Colleen, Hongling, Lila, Saray, Galina, Chuling, and Iwa. And then Ben Soroff in Vienna have recently left, and my collaborators, Teresa Dunn and Cindy Tift.